We have talked about all three domains of life: bacteria, archaea, and eukaryota. And within the domain eukaryota, we have talked about three kingdoms: protista, fungi, and plantae. Today, we talk about the last kingdom, Animalia. To review, bacteria and archaea are special because they have special prokaryotic cells with no nucleus or other membrane-bound organelles. Protists are unicellular, like bacteria, but are eukaryotes without cell walls. Fungi and plants are both eukaryotes, are both mostly multicellular, and both have cell walls. Their cell walls are made of different materials: peptidoglycan in bacteria, chitin in fungi, and cellulose in plants. Another difference between fungi and plants is that fungi have only one type of tissue, the hyphae, whereas plants have many tissue types. Lastly, plants are photosynthetic, so we call them autotrophs, whereas fungi eat other organisms, so we call them heterotrophs. So what are animals? Well, that is what today is all about. Of the organisms shown in this photo, which ones are animals? My hint is that anything not in one of the other groups we have covered would be an animal. All animals have eukaryotic cells. They're multicellular with many tissue types. Their cells do not have a cell wall. Animals have a unique protein called collagen that glues together connective tissue. In mammals, it makes up about 30% of all proteins in the body. It is responsible for tightening the skin, and as it degrades with age, we develop wrinkles. One way to reverse the process of getting wrinkles is by injecting the skin with more collagen. Collagen was literally used as the first glue 800 years ago, through melting horse cartilage and skin. This is also how gelatin is made. Many animals have the ability to move at least during one stage of their life, unlike fungi or plants that are stationary. We refer to organisms that do not move as sessile. Sea anemones and giant clams are sessile as adults, but have larvae that can swim. Seeds and spores, while they do get moved around, can't really move much on their own. We used to think that all animals were heterotrophs, eating other things, whether they are herbivores, carnivores, a parasite like this tapeworm, or a scavenger like this vulture. But we have recently discovered that a few animals have evolved to be solar-powered, like plants. The ancestor of this sea slug ate algae and stole their photosynthesis genes so they no longer need to eat anything. The spotted salamander lays its eggs in algae, and they have a symbiotic relationship where the embryos gain energy from the algae. In biology, exceptions to the rule like these are common. Over the next few months, we will be exploring the kingdom Animalia in great detail. Animals are broken down into 33 phyla or main groups. This phylogeny displays groups in terms of their relative size, as well as showing when they branch off from other groups. We are going to focus on the most important 9. You should be at least a little familiar with these groups. I'm sure you've heard of sponges, flatworms, mollusks, and arthropods, but I'm going to tell you about their ecology and evolution because we can now use your knowledge of these topics to better understand the diversity of life. We will cover these groups in 2 separate units: invertebrate diversity, which will include 8 phyla, and chordate diversity, which will cover just 1. We will spend more time on chordates because they vary so much in form. Chordates include fishes, amphibians, reptiles, birds, and mammals. Animal diversity is striking. Sure, trees and cacti look kind of different, but worms, beetles, jellyfish, and frogs are really, really different in terms of how they look, move, eat, defend themselves, and reproduce. This should make you question why. Why are animals so diverse compared to everything else? Our planet is 4.54 billion years old. The first life evolved approximately 3.7 billion years ago, based on what fossils we have been able to find and date. Bacteria ruled the planet for almost 2 billion years. According to this diagram, when did the first protists evolve? It wasn't until 640 million years ago that the first animals formed in the oceans. Plants did not evolve until 470 million years. Our species has only existed for a mere 300,000 years, a blink in time. Notice something about this phylogeny. Most phyla evolved at around the same time, 451 million years ago. Something drove animal diversification to occur very quickly and create a variety of very different body plans. This explosion of animal body types is called the Cambrian explosion. This video explains what we think happened and what life was like during the late Cambrian period.
When the clock of animal evolution began to turn, life on Earth seemed simple, sedentary, and peaceful. Then in a flash, everything changed. Over 500 million years ago, fantastic creatures sprang into being as though from nowhere. In geologic time, these animals appeared in the blink of an eye. For the evolution of life on our world, it was a singular, defining moment. Des Collins has dedicated his career to the pursuit of these ancient creatures. As a paleontologist, he wants to look back in time. All the way back to more than 500 million years ago. By peering into the darkest recesses of animal history, Collins hopes to shed light on what has long remained a mystery. At Toronto's Royal Ontario Museum, Collins works with a vast repository of fragments from the ancient past. Fragments that reveal a world unknown until recently.
We have a much better one over here. There's the claw, the large predator. Oh, yeah. It shows up much better. Yeah,
These fossils from high in the Canadian Rockies form pieces in a puzzle that tell the story of early life on Earth.
Yeah, that looks great. Thank you.
Okay, good, good.
The locality I work with is what's called the Burgess Shale. It's a very famous locality, known since it was first discovered about 1909. The numbers of fossils are in the tens of thousands. The first person that collected the famous locality out west in Western Canada around the turn of the century, he collected something like 65,000 specimens in 5 seasons.
Strange, almost unearthly creatures were discovered in the Burgess Shale. They existed over 500 million years ago during the Cambrian period, 8 times earlier than the last days of the dinosaurs. But more astonishing than their antiquity was the way these bizarre animals appeared, as if all at once, suddenly coursing through the world's oceans. Evidence of this unique event is not isolated to the Canadian Rockies. Similar sites have been uncovered all over the world. This extraordinary burst of animal life was dubbed the Cambrian Explosion. If we could go back in time, before fish, before complex animals roamed the seas, and before the Cambrian explosion, we would find only simple creatures. The course of life was relatively slow. Sponges, flower-like organisms, and flatworm-like animals dominated the ocean realm. Then, evolution took an abrupt turn. Suddenly, wondrous new life forms filled the watery world. A marine worm called Achaea was remarkably complex and mobile. Survival depended on competition for food, and the ocean became a realm of hunter and hunted. The Cambrian was a time full of sudden danger. Even in such a savage world, Animalia stood out. Nothing matched it for its sheer size and killing efficiency. Snaring its prey with its 2 powerful front claws, it was a swift and deadly hunter. Only slightly lower on the food chain, another fearsome predator prowled the oceans. Opabinia had 5 eyes at the top of its head and its own grasping claws to snare the weak and unsuspecting. To counter such ferocious predators, animals adapted new defenses. Canadia, a worm-like creature, sported protective bristles along the length of its body, essential armor for a slow-moving beast. Wiwaxia also glided on the ocean floor, depending on their elongated spikes for protection.
As the video states, the evolution of predators caused an evolutionary arms race that likely drove evolutionary change. But this was not the only cause of the Cambrian explosion. During the same period, oxygen levels began to rise because of the photosynthetic cyanobacteria, which allowed life forms to grow faster and larger. Thus, a constraint on evolution was removed. Lastly, we believe that animals evolved special genes during this time that were master controls of other genes. These are called Hox genes. An example is a gene that controls wing development in insects. A simple change to one of these Hox genes can lead to multiple wings, limbs, or body segments, thus allowing a greater range in possible body forms. In order to compare animal phyla, you will need to know something about how they develop. So I want to define some terminology that will be helpful in the next unit. Animals can be defined based on their symmetry. The first animals, sponges, have no symmetry. They have random shapes and cannot be cut into 2 equal and identical parts. Other animals are like pizza pies. They can be cut into 2, 4, 6, or 100 equal slices. We call this radial symmetry. Humans have evolved a third type called bilateral symmetry. We can only be cut into 2 equal parts. Let's take a look at development in animals. All animals start off as a single diploid cell called a zygote, which is an egg cell that has been combined with a sperm cell. The cell divides and divides until it forms a hollow ball called a blastula. Then something important happens. A tube starts to form in the middle, as if someone stuck their finger into a soft ball of clay. This process is called gastrulation, which means formation of the gut. The embryonic stage where the gut is forming is called the gastrula. The big question is, will this opening be the mouth or the anus? The answer depends on what animal we're talking about. If they form the mouth first, they're called protostomes. Most animals are protostomes. If the anus forms first, they're called deuterostomes. Vertebrates like us and echinoderms, sea cucumbers, and sea urchins are deuterostomes. Is it really a surprise that we start life as little buttholes? Furthermore, realize that you are really just a donut with a tube running through your middle. If you eat a penny, does it ever really enter your body? No, it eventually falls out the other end. Food only enters the body as molecules tiny enough to pass across the gut lining into our bloodstream. Something else important is happening during this phase of being turned into a donut. Different tissue types start forming. All of your tissue is derived from 3 main types, called germ layers. The ectoderm is on the outside and turns into our skin. It also forms our nerves, which is obviously not on the outside of our body, but this is another story. The endoderm is the layer that lines the tube that becomes the lining of the gut and lungs. The last layer, the mesoderm, becomes everything else. At the gastrula stage shown, which tissue layers have formed? The next stage in development is to form the mesoderm, add jelly into the donut, and complete the formation of the tube. Notice that the mesoderm is hollow. The opening is called the body cavity or coelom. You can see it clearly in this dissected piglet. Your lungs, heart, stomach, liver, and other organs don't sit in a pool of blood or fluid, but hang in an empty body cavity. The formation of this body cavity will be important in later lectures because it forms in different ways in different phyla. From there, the ball of cells continues to divide and grow. Organs are formed through a process known as organogenesis, and amazingly, from a single cell forms a multicellular embryo that will eventually become a complex life form, independent of its mother. Stage 22 on this diagram takes 54 to 58 days, and at this stage, the embryo is the size of a blueberry. I wanted to show you a time-lapse video of a salamander developing from a zygote to a larvae. You can see the formation of the gut very well in this video. You can see the embryo becoming bilaterally symmetric. This takes place over a 3-week period. Frogs can develop in as little as a single day if they are in warm water. You can see blood circulating through its translucent body. You can see its beating heart, the development of the eyes and the gills, and eventually the stored food in the egg is used up and the larvae hatches. It is now ready to find food and grow into an adult capable of finding a mate and starting the process all over again with its offspring.